Welcome to the Lundbeck webinar on brain circuits for spatial navigation. This is one in a series of 6 webinars that are available for view on the Lundbeck Foundation website. We have 3 people who've really done fundamental foundational work in the area of spatial navigation today. First, Anne Hermundstad, who is a group leader at the Janelia Research Campus and investigates how the brain creates and uses adaptive sensory motor representations to generate flexible behavior in the face of uncertainty and environmental change. She uses a combination of theory, modeling, and data analysis in close collaboration with experimental groups. Our second speaker will be Ila Fiete. She's a professor in brain and cognitive science and the director of the K. Lisa Yang Icon Center at the Massachusetts Institute of Technology. Ila's work focuses on how neural ensembles support learning, integration of information, inference, and spatial navigation. She uses, develops, and adapts approaches from mathematics, machine learning, and physics. And finally, our third speaker is Barbara Webb, professor of biorobotics at the University of Edinburgh. Barbara's work combines theory, experiments, and robotics to study how neural systems control behavior. Her research includes studies of multimodal integration, navigation, and learning, particularly in foraging insects. So our first talk is from Anne, and the title is Induction Biases for Rapid Learning in Spatial Navigation. Beautiful, beautiful.
Beautiful. Okay. Wonderful. Fourth time is a charm. Thanks so much, Larry, for the introduction and for having me here. I'm really excited to tell you all about work that we've been doing to understand how different animals, from insects to mammals, flexibly navigate through their surroundings. And most of the work that I'll be telling you about today was done in close collaboration with Vivek Jayaraman's lab at Janelia in fruit flies. But this work has also been informed by discussions and more recent collaborations with Josh Dudman's lab in insects. So we've been interested in understanding not only how animals navigate through their surroundings, but also how they flexibly modify their behavior in response to changes in those surroundings. And so to get at these types of questions, we've been studying a range of different navigational learning paradigms where animals have to physically or virtually move through their surroundings in order to gather rewards or avoid punishments. And so in trying to understand animal behavior in this setting, we've been particularly interested in understanding to what extent this behavior reflects hardwired versus flexibly learned components. And so what I hope to tell you about today is how behavioral flexibility, which we often think about in the context of online learning, can be enabled by hardwired neural circuit architectures, what we often call inductive biases, that enable this learning to be fast and efficient. So one way that we try to understand how animals learn in these types of settings is by designing artificial agents that try to mimic the behavior of real animals. And in these types of navigational settings, a very common way to build an artificial agent is to assume that it lives on a 2-dimensional grid and that it navigates this grid using one of a discrete set of actions. So stepping forward, backward, left, or right. And if this agent doesn't have any initial preferences in terms of which action it likes to take, then when it first enters into this new environment, it will diffuse randomly on this grid. And if we're patient, we'll see that this agent will eventually stumble upon this hidden target and will get a reward. And when that happens, the agent can learn that something about its past behavior led to its current success, and it can reinforce that something. And a simple thing to reinforce is the action that the agent just took right before getting a reward, which in this case is stepping forward from this particular grid location. And the agent can store that information in a lookup table that keeps track of how valuable it is to take each of these 4 actions from each of these different grid locations. And so we can watch that lookup table building up over time as the agent learns to navigate towards this hidden target. And that's what I'm going to show you in this simulation on the left. So if we allow this agent to continue exploring this environment, we see that it initially stumbles upon the target from different orientations and initially learns which actions to take in the immediate vicinity of the target. But over time, the agent learns that certain actions lead to other actions that lead to the target, and it learns to reinforce these chains of actions. And we can see this in the diffusion of this color map backwards from the target toward the starting location. And this is going to guide increasingly straighter trajectories from the start to the target. Now, as I mentioned, this is a very common algorithm for modeling navigation and navigational learning, but it has several important drawbacks. So one of these is that this algorithm is incredibly slow. So here the agent has to associate 4 different actions with a 30 by 30 grid of locations. So this is several thousand parameters that it has to keep track of in principle. These algorithms don't generalize well. So if the agent starts in a new part of the environment, it suddenly doesn't know how to get to the target, even though it's been there in principle several hundred times before. And finally, these algorithms generate behavior that doesn't mimic what we see in real animals. And so we've been interested in moving beyond these types of algorithms to better capture the structure that we see in animal behavior, better explain how animals are able to very rapidly modify that behavior in new settings, and ultimately understand how these algorithms are implemented in neural circuits in the brain. So one simple way that we can move beyond this type of picture is by designing agents that come pre-equipped with some behavioral structure from the onset. So for example, we could design an agent that navigates this environment in loopy trajectories rather than in these elemental actions. And then we could reframe the learning problem in terms of scaling and shifting this loop rather than building it up from scratch. And Josh Studman's lab recently showed that this type of description better captures how animals behave in a hidden target foraging task and better capture how animals are able to very rapidly modify their behavior to new target locations compared to these sort of classic tabular learning methods. So what I want to tell you about today is how we've been taking a similar type of approach to understand behavioral flexibility in a smaller creature, the fruit fly, and how we can use the fruit fly to get a deeper understanding of the neural circuit mechanisms that might underlie this behavioral flexibility. So with Vivek's lab, we've been studying a much simpler variant of this navigational learning paradigm where flying flies navigate in a virtual one-dimensional arena to gather rewards or avoid punishments. And just as we did before, we can build an artificial agent that now navigates on a one-dimensional grid by stepping to the right or to the left. And this agent over time will learn that at some grid locations, at some headings, it's better to turn to the right, and at other headings, it's better to turn to the left in order to reliably hit this target. And again, we can consider a second agent that comes pre-equipped with this behavioral structure from the onset. So this second agent already knows how to structure its turns relative to some central heading and needs only learn to shift and scale this function in order to align it with this target. Now, the second agent on the right learns much more quickly than the agent on the left for a few different reasons that I alluded to earlier. So the first is that there are just far fewer parameters to learn. And the second is that this agent on the right can immediately generalize what it learns at one location and apply that to other locations. And so to see how that could work, we can consider a scenario where these two agents are at the same heading when they receive a reward. The agent on the left will respond to that reward by adjusting the action it takes at its current heading, whereas the agent on the right will learn by shifting this entire function, which changes the action it takes at its current heading and also changes the actions it will take at all other headings, even if it hasn't visited those headings before. And so this ability to immediately generalize what you learn at one location and apply it to other locations can significantly speed the learning process. Now, for this to be the type of algorithm that flies use, this would imply that the brain would have to build in this type of behavioral function, and there would need to be neural circuit mechanisms that could shift and scale this function based on experience. So I want to tell you how we think that could be done. But first, I want to give you a little intuition for how this could work in principle. So let's consider a simple scenario. So I'm here in my office, and let's say that I want to head over to my sofa, and I need to figure out which direction I need to turn. So one simple way that I could figure that out is I could imagine turning to my left, and I could imagine turning to my right, and I could ask which of these two possibilities overlaps with where I want to go, the sofa. And in my case, it's this possibility on the left, which tells me I would need to turn to the left in order to get there. Now, to implement an algorithm like this, we would need to be able to keep track of where we are right now, our heading, and I'm going to represent that by this white vector here. And we would need to be able to imagine turning to the left, which you can think of as rotating this vector 90 degrees to the left or to the right. And then we need to compare these two possibilities with a memory we have of where we want to go. And I'm going to call this our goal. And whichever has the greater overlap tells us which direction to turn. And I'm just going to denote that overlap with these saturated color maps here. So indeed, we see that the left-hand scenario has the greater overlap and tells us we need to turn to our left. Now, perhaps it's easy to see how this algorithm works if your goal is directly to your left or directly to your right. But in fact, this algorithm can work regardless of which direction you're facing relative to the goal. And so to see that, we can repeat this calculation, varying our relationship to our goal. So I'm going to keep my goal fixed. The scenario that I just described is when my goal is to my left, and I'm going to rotate my own heading away from that goal. And so if I rotate away from the goal, at some point, I'm going to be facing directly opposite the goal. And you can see that there's equally weak overlap between these two possibilities, which tells me I could turn in either direction to get there. If I continue rotating, at some point, my goal is going to be over to my right. And this, again, will now tell me that I need to turn to my right to get to that goal. And so if we sort of sweep through this entire set of possibilities, what we've done here, sort of rebuilt this behavioral function that we derived earlier through completely independent methods, through reinforcement learning methods. And this is what we would call a behavioral policy that specifies what actions we should take as a function of where we are right now relative to where we want to go. And now if I change where I want to go, I decide I want to head over to my whiteboard instead of to the sofa, I need only update this memory of my goal, and all of this behavioral structure will come along for free. So in other words, if I change where I want to navigate to, I don't need to relearn how to navigate, I just need to redirect my navigational program to that new heading. So we've sketched this out using simple intuition and these simple schematics, but how we arrived at this was deriving this mathematically, working backwards from some specification of what we ideally want. So we derived this function through reinforcement learning methods, and we posited that it would be smart for the brain to build this function in, and we asked how might it be possible to do this using simple neural circuit-like operations. And so you can work backwards and suppose that you have an internal representation of your heading. You can apply a phase shift to that heading and multiplicatively combine that with some representation of a goal in order to compute this overlap. And now you can show mathematically that you are guaranteed to get this behavioral structure at the output by supposing these particular computations, regardless of the properties of the goal itself. So this means that you can flexibly change and modify the goal, and the behavioral structure that you have at the output will always retain this same shape. So now we're going to dive in and see how this might be implemented in the brain. But I just want to highlight here that this builds on really important work from Barbara and Stanley Heinze from several years back. Okay, so this is how the brain could, in principle, structure behavior relative to where you are now versus where you want to go. And so I'm going to take you into the fly brain and tell you how we think this is implemented. And we think this is carried out in a set of central brain structures called the central complex, shown here. And I'm going to expand these structures and take you through them one by one. So starting with this region at the top. Now, we know from previous work that this donut-shaped region maintains an internal representation of the fly's heading. And this is maintained in a population of so-called compass neurons that innervate this donut in wedges. And if we image calcium activity from this population of neurons, we can actually see this internal representation of heading. And this is what Johannes Seelig discovered when he was a postdoc in Vivek's lab. And so here he's looking at the activity of this whole population of neurons while a fly is navigating in a visual virtual environment. So the fly's head is fixed in place. It can walk on a freely rotating ball, and the rotations of that ball are going to drive rotations of a visual scene in front of the fly. So if I play this video, you can see the visual scene moving in front of the fly, and this activity forms a localized bump that tethers to the visual scene and tracks the movement of the scene relative to the fly. So we think of this bump of activity as the fly's internal compass needle that maintains a representation of which direction the fly is headed relative to its surroundings. And now we can follow this heading representation downstream into these downstream brain regions based on what we know from the electron microscopy connectome about the neurons that interconnect these regions. And so to help us with this, I'm going to color the wedges of this donut according to the phase of the heading bump, its orientation. So if the fly is orienting in a particular direction, one of these wedges, like you see here in orange, will be active. And now there are neurons that carry this activity to two localized regions in this bridge-shaped structure, and another population of neurons that carry this activity to a single localized column. in this fan-shaped structure. And there's an entire population of these neurons that repeat these projection patterns across these sets of structures. And so we can use this to essentially propagate this phase coloring downstream. So you should think of this as an anatomical reference frame that's anchored to the fly's current compass heading. And now there are other neurons that perform operations in this reference frame. So for example, there are neurons that project from orange to yellow. And this corresponds to what we described earlier as imagining turning to the left, although now we don't have to invoke any notion of imagination. This corresponds to taking a bump of activity that represents the fly's heading and shifting it 90 degrees to the left. And there's another population of neurons that shift this bump of activity 90 degrees to the right. And these two populations project to premotor regions that are involved in initiating rightward and leftward turns. So we think that these neurons are well positioned to guide the fly's actions based on where the fly is right now. But there's one additional piece we need, and that's where the fly wants to go, the memory of its goal. And we think that this goal is stored in the strength of synapses of neurons that innervate layers of this structure. So putting these pieces together, we think that this architecture could guide behavior now based on where the fly is relative to where it wants to go. And indeed, recent physiology work from Gabby Mayman's lab and Rachel Wilson's lab is consistent with this picture. So here you can see calcium imaging from the output of these phase-shifting neurons in these premotor structures that shows that you get this sinusoidal structure that you would need to guide the appropriate turns based on where the fly is relative to its goal. So there's one last step I want to talk about with this, and this is that in principle, we'd like to be able to shift this goal based on experience. And we think that that's done through a form of Hebbian-like plasticity that's modulated by dopaminergic neurons that innervate the same layers of this fan-shaped structure. And from the connectome, we know that these dopaminergic neurons themselves receive information about the fly's current compass heading. And so that means that in principle, they could modify the strength of these goal synapses in a heading-dependent way. And so the way that we think that this could work is that these neurons could imprint a bump of activity on the goal synapses based on whether the fly is getting rewarded or punished at a particular heading. So if I'm orienting a certain way and I'm getting rewarded, I can imprint that bump of activity on my goal, and that will pull my goal heading towards where I am right now, where it's currently good. And so this could be used to modify the strength of these goal synapses and shift the goal based on experience. And as we said before, all of this behavioral structure will come along for free based on this hardwired anatomical architecture. Okay, so stepping back and putting these pieces together, I've told you how we think the fly guides its actions based on internal representations of where it is relative to where it wants to go. And I've told you that we think that this heading representation can be used to build and flexibly modify this goal representation over time. And of course, this is unfolding in closed loop, which means that the fly's actions will be changing its heading and the development of its goal over time. Now, this all assumes that the fly has an accurate internal representation of heading that is accurately anchored to the outside world. But we also know that this heading representation itself is flexible and is flexibly tethered to different cues in the environment. And so in the last minutes of this talk, I want to work upstream and tell you a little bit about what we know about how this heading representation is formed and how it's modified. Okay, so we're going to move upstream. As I mentioned before, this internal representation of heading is maintained as a localized bump of activity in a population of so-called compass neurons that innervate this donut-shaped structure. And one of the great successes of recent fly neuroscience has been our ability to understand how the fly circuitry, these compass neurons and other neurons in this structure, could accurately maintain and update this bump of activity as an accurate compass needle. And this builds on decades of past theoretical work that tells us how the brain could, in principle, implement a compass needle. And now we're at the point of being able to test some of those ideas mechanistically and understand how they're implemented in the fly brain. And so one of the things that we've been interested in understanding is how a circuit even as small as a fly brain could do this very accurately by precisely tuning the strength of local excitation between these compass neurons. So we know from past work that this compass signal can be updated purely based on self-motion signals based on the fly's turns. And so this allows the compass needle to update even in complete darkness. But this compass also has to flexibly tether to different sensory cues in the environment. And we know that this information about localizing sensory cues comes in through another pathway, another set of neurons called ring neurons, one of which you see here in blue. And these ring neurons bring in information about complex visual features, mechanosensory features like wind, and polarized light cues. Now, these different ring neurons make all-to-all connections onto the compass neurons, one of which you can see here, but not all sensory modalities are treated equally. And this was something that was discovered by Hannah Haberkern, who was a postdoc in Vivek's lab at the time, and Marcella Norman, a postdoc in my lab, when they were analyzing the connectome. And they discovered a potential hierarchy in terms of the different sensory modalities that tether the compass based on the number and location of synapses that these ring neurons make onto the compass neurons. And so this suggests that the hardwired structure of the fly's compass prioritizes some sensory modalities over others. And we think that this prioritization reflects how reliable these different sensory modalities are in generating compass cues in natural settings. For now, I'm just going to focus on one particular modality, complex visual cues, and ask how the fly keeps its own compass in register with these external cues in the environment. Now, these complex visual cues come in through a subset of visually responsive ring neurons that have local receptive fields that see different regions of a visual scene. And there are many of these ring neurons whose receptive fields tile visual space. And now we can start to appreciate the challenge that the compass faces. So when the fly is navigating in a visual environment, it's receiving inputs both about the fly's own self-motion and about these localizing visual features in the outside world. And to keep these two sets of cues in register, we think the circuit relies on a form of activity-dependent plasticity that modifies synapses from ring neurons onto compass neurons when they're coactive. So to show you how this could work, I'm going to unfold this donut here so that our compass neurons are arranged vertically and our ring neurons are arranged horizontally. And I mentioned that these ring neurons make all-to-all connections onto the compass neurons. We know that these connections are inhibitory, and so we can represent the strength of that inhibition by the size of these circles. And I'm going to consider a simpler visual scene than the one you see here with just a single visual bar. And this visual bar is going to activate one of our ring neurons that you see here in blue. And at the same time, the bump of activity is going to be somewhere in the compass. I'm going to assume it's maintained by this compass neuron here in white. Now, the coactivity between these two neurons is going to weaken the synapse between them. And you can see that by this shrinking circle here. Now, if the fly turns, the visual scene will move, activating a new ring neuron, and the compass bump will move, activating a new compass neuron. And again, the coactivity of these two neurons is going to weaken the synapse between them. And if we carry this logic through, we see that over time, this plasticity will develop a self-consistent mapping between the visual inputs that drive the compass and the self-motion inputs that drive the compass. And this is represented by this band of weakened inhibition along the diagonal of this weight matrix. So this shows how the compass could, in principle, keep these two different sets of inputs in register. But is this actually how it works in practice? Well, if we monitor the bump of activity while the fly is navigating in this same visual scene with respect to the single bar, you can see that as we saw before, the bump and the scene move together in lockstep. And this reflects this self-consistent mapping on the left. But if this mapping is created through activity-dependent plasticity, that means we should in principle be able to alter this mapping by manipulating the activity in the compass. And that's exactly what Sung-Soo Kim, who was a postdoc in Vivek's lab at the time, set out to do. So in one of the strongest tests of this idea, Sung-Soo tried to invert this mapping, and he did that by optogenetically activating a bump of activity in the donut while rotating the visual scene in the opposite direction. And he did this in open loop. And then when he gave the fly back closed loop control of this visual scene, you can see that this mapping did indeed invert. Now the scene and the compass bump move in opposite directions when the fly turns. Now, this will revert to the original mapping over time based on the same plasticity mechanisms, but this tells you just how flexible this compass circuit is. And in more natural settings, we think that this plasticity allows the fly to tether its compass heading to a range of different sensory cues in the environment. And we in other labs have been interested in understanding at a more mechanistic level how this is implemented. Okay, so stepping back and putting everything together, I began by telling you how the fly could modify its behavior based on where it is relative to where it wants to go, and that this internal representation of heading is itself informed by changes in the sensory environment through these incoming sensory channels. And these two pieces of this story that I've told you sort of highlight two different learning systems that we think are involved in flexible navigation. One that's involved in mapping external cues onto this internal representation of heading, and a second that's involved in using this heading representation to build and modify goals. And these two different learning systems involve a combination of both hardwired and learned components. So we saw that the compass itself has a hardwired prioritization of different sensory modalities that tether the compass. But within the constraints of this prioritization, we think that the compass bump can flexibly tether to different cues in its environment. And we think that this unfolds through an unsupervised learning process. Moving downstream, the compass heading is used to build and update goals and drive behavior within the constraints of a hardwired behavioral policy. So we think the form of this behavioral policy is hardwired, but the parameters of that policy, namely the goal heading, can be flexibly modified based on experience. And we think that this unfolds via a reinforced learning process. And so one of the things that we've been interested in exploring recently is how these two different learning systems interact and work together when flies explore and learn about new sensory environments. But perhaps taking a bigger step back, I think one of the lessons that we're continuing to learn through this work is that hardwired neural circuit architectures can enable rapid behavioral flexibility by exploiting certain predictable structure in the environment and in the consequences of an animal's actions in that environment. And I think that this predictable structure gives us a lens for asking what might be similar or might be different across different species that we're studying. So if certain features of the environment are so predictable that every animal has to contend with them, the brain could benefit by hardwiring those features in, and we might expect to see similarities in the neural circuit architectures that we see across different species. So I think this gives us an axis to ask, what should we expect to be similar and what should we expect to be different? So with that, I just want to thank the fantastic lab that I work with and give a huge thanks to Chuntao, Ramya, Brad, and Vivek, and their work is what I highlighted today. So thanks.
Thank you. Yeah, maybe I'll start off the questions, but everybody's welcome to ask. So you had this very nice interpretation of the sinusoidal steering curve as kind of a generalization strategy. So is it an optimal generalization strategy? Is there a way to show in some situation that that's an optimal curve?
I think it depends on what the goal is. So we derived that curve through reinforcement learning methods, assuming that the fly needed to maintain a single goal heading. But if you had other goals that you used to optimize that policy, you would get out different behavioral curves. And so the degree of generalization depends on how well aligned that policy is with the task that you're now trying to solve. So we could certainly introduce other tasks that would break that policy where you would make the wrong generalization. So I think that depends on what we think, the function of the animal or the circuit is in a setting, and that would allow us to make statements about whether that gives you optimal generalization for the class of tasks that would represent.
So then, do you think that curve is adaptable?
Yeah. So we're supposing here that the curve itself, the shape of the curve is fixed, and the amplitude and the offset are adaptable. But it's an interesting question to ask, could you then, perhaps over longer timescales, actually modify the curve itself? Behaviorally, we haven't been able to push that axis and train flies in longer settings to see whether they could really refine their behavior and change it over longer timescales. But yes, I think this comes down to sort of a hierarchy of different timescales that could be used to build in different components of these functions. So we might think about ordering different aspects of those functions that could be flexibly changed.
Okay, Barbara, Ila, you have any questions?
One thing I've been thinking about quite a lot lately is that we've got very nice descriptions now of how the animal might alter its heading to align with a goal. In other words, steer towards something. But insects in general don't always face the direction they're going. So, and I'm going to talk a little bit about that in mind, but I'm thinking I'm not going to talk about the output side of that, if you like. So I still have a steering system at the end of the models that I'm working with. But do you have any thoughts on how you might generalize so that the output is not turn left or turn right? But
yeah, so actually I glossed over a lot of what we've been thinking in that domain. So we've been starting with 1D flight behavior where flies are fixating and saccading. So they're not continuously steering toward a goal. And we've looked at both of those modes in flight. And you can derive using the same types of approaches what the optimal policy would be for controlling fixations. And the steering policy that I was showing you, we've actually been thinking about in the context of controlling the probability of turning to the right or to the left. So it's not necessarily that you are continuously steering towards it, but it tells you how to proportion your actions depending on where you are relative to that. And so I think this type of approach could be generalizable to the different modes of behavior that we see. And our way of thinking about it has been to take the behavior itself and try to decompose it into these different modes, look at the statistical structure of that, and then try to ask how that structure could be modified online. And so as we move to other settings where there's sort of more behavioral variability, I think that would give us an access to sort of expand this behavioral policy to include sort of richer modes of movement.
Okay.
But yes, in short, we think that this other class of neurons, the PFL2 neurons, is actually controlling the forward flight in a way that's very consistent with these same ideas.
I actually had a very related question. I mean, when we talk about flexibility from the mammalian perspective in navigation circuits, we see that the same circuit may be used for navigating the space in different ways, with physical movement or, observational sort of exploration. And I was wondering if there's an analog to any of that in insects. And so it seems like that's certainly it's certainly the case that the same circuit can be used to direct different types of movement. It can also receive different types of inputs to, tether or otherwise, track head direction. Do we have clues that these animals maybe reuse that circuit in very different behaviors in other ways?
Yes, and I think Barbara will talk about some of that. But I guess one aspect of your question is whether flies could mentally navigate. So could you generalize? And I don't know that we have any evidence of that in the way that's been studied in mammals. But the fan-shaped body receives lots of inputs from the mushroom body. There are all sorts of these tangential neurons that we think are involved in maintaining information about different contextual cues. So I think a big open question is how these different types of inputs are mediated and arbitrated between to make decisions about certain behaviors in certain contexts. But certainly the fan-shaped body is a very rich recurrent structure with lots of contextual input and the possibility of driving different types of behaviors based on that input. And I think there's a question of how the fly makes decisions in the face of really rich sensory input when there's lots of cues coming in that could potentially guide behavior.
Okay, thanks. I think we'll move on to Ila's talk. Our next speaker is Ila Fiete. Her title is Mammalian Head Direction Circuit Dynamics and Comparison with Insects.
Thank you, Larry. Let me fidelity mode. I hope you're able to see the screen.
Okay, yeah, that looks good.
Okay, fantastic. Well, thank you for inviting me to join this exciting panel, and my heartiest congratulations to the Brain Prize winners for this year's Brain Prize. I think it is just fantastic and very validating for the computational neuroscience community as a whole to have, three people who are role models in the field of computational neuroscience receive this acknowledgement and this honor, and to also take stock of how computational neuroscience has shaped our understanding of computation, dynamics, and structure in the brain. So it's a great honor to be able to tell you a little bit about what we understand about circuit dynamics in the head direction system. In particular, I'm the outlier in this panel. I'll be speaking a little bit about our investigations in mammals and specifically in the mouse. So one of the most interesting things that jumps out in thinking about the difference in head direction circuitry between insects and mammals is this notion of spatial organization and spatial topographic order in the head direction circuit. So, of course, Barbara, I think, is going to be talking about a lot of exciting work in the direction of comparative circuitry across insects. And in fact, I've taken this slide from Barbara's paper, in which what we see here is we see that the fruit fly has the famed physical ring that underlies the dynamics of head direction dynamics in the fly brain in this ellipsoid body, this donut-shaped structure. In addition, of course, is the protocerebral bridge, which is bidirectionally coupled to the ellipsoid body, and there's the noduli that bring in velocity information that then drives asymmetries in these circuits to update the state of the heading estimate in the circuit. So what you see on the left here is that there's clear not only physical anatomical correlate of a ring of states, but also this physical ring. And in fact, there's a lot of beautiful topographical order at the level of single neurons even. And in the middle here, I'm trying to give you a depiction of what we understand about a comparable circuit in the mouse. And so on the very top of the central panel here for the mouse slide, you can see that there is a set of different areas, multiple different areas that receive sort of on the bottom, we see vestibular inputs coming in, and then in green are the circuits that are responsible for encoding head direction. And the belief is that these two areas, LMN and DTN, generate the head direction signal and then broadcast it into this area ADN, which is the thalamic nucleus, which then in turn broadcasts the signal to cortex. And the question about whether there is spatial order and spatial topography in this circuit, I think is very much up in the air. So recently, beautiful experiments from Mark Brandon's lab did some imaging directly of this ADN nucleus, and color-coded neurons according to their relative head orientation preference. And at the bottom here, you can see this plot, and it's very difficult to perceive any kind of order, either in the sense of the structure of the whole circuit being in a physical ring, and also if you look at neurons and their somas and their orientation tuning, you can see that there does not seem to be a co-localization of co-tuned neurons in the physical space. So what are we to make of the extreme correspondence between physical and spatial structure in the fly and this apparent complete absence of physical topography in the mouse? And I think what's interesting is this example of the locust, where these highly conserved structures that nevertheless code for head direction, like the ellipsoid body, now have a slightly different form in the locust, where the ellipsoid body doesn't close back on itself in this physical ring structure. So it seems like anatomical topography is certainly a very compelling type of order that is present in the fly system. It's present in a much greater degree in another insect like the locust, and it seems to be quite entirely absent in the mouse. And I think this gets to some of the fundamental questions about topography in general and brain maps across species. And what is the relationship between physical order and function. So here are a few more examples taken from a recent review of different insects and their ellipsoid bodies. So just like the locust, the honeybee also does not have a fully closed ellipsoid body, and the Madeira cockroach, though it has a similar overall central complex organization, but once again, their ellipsoid body is not closed. All right, so now, unfortunately, because as you can see from the previous slide, we don't have quite the luxury to characterize the detailed anatomical order of the mammalian head direction circuit. We do have the luxury to, in a similar way as the fly, to look at the dynamics in the circuit. So first, let's look at the dynamics in the fly head direction circuit. And of course, this is beautiful experiments, in this case from the Jayaraman lab at Janelia. And here, now famous, we have this video of this the bump of activation within the ellipsoid body moving around as the fly is moving tethered on this virtual reality setup on a ball. And you can see here the angular estimate of the fly's direction as it's moving, as well as the actual pattern position of the visual stimulus that's shown to the fly. So this is beautiful. So we've got this bump of activation that's tracking the instantaneous heading direction of the fly, and here is a depiction, just a stationary depiction of in a graph of the position of the bump over time. And we can look at in the mouse, we can look at how the circuit is, how the states in the circuit encode head direction. And so this is just a stationary snapshot of the states of the circuit of about, in this case, about of order 1,000 to 2,000 neurons in this area ADN. And you can see that the states lie on this, quite beautiful one-dimensional ring structure, although the one-dimensional ring doesn't seem quite entirely flat, partially due to sampling issues. Now we can look at the dynamics of the states on this one-dimensional ring. So this is a projection of that of the ring into 2D, and then what you're seeing here is in purple is the trajectory of states as the animals moving around, freely exploring in a two-dimensional environment, and you can see that the states are tracing out different trajectories on this one-dimensional ring structure. So although we don't have the same anatomical organization, but the mammalian system has beautiful dynamical order that seems similar to the, we can by looking at the states in the state space view, we can look at kind of, if not the evolution of one activity bump, but we can look at the evolution of the whole population along this one-dimensional ring structure. So the thing that was exciting as a theoretical neuroscientist in the heady days when we're able to do large-scale population recordings is to take models of the head direction circuit that have existed for 20 or 30 years and try to understand to what extent the dynamics predicted by these models is matched by the dynamics in the head direction circuit. And so the head direction model makes some fundamental dynamical predictions in addition to, of course, the structural predictions, which are not yet able to be tested in the absence of a connectome in the mammal. So let's consider some of these fundamental predictions over here. And so what we have is we've got, I've kind of, written down 5 core predictions of the models. So the first prediction of models in general that build of models that try to explain how the brain is capable of representing a continuous variable, like, for example, the head direction variable of angle from 0 to 2 pi, and then is also capable of using velocity inputs to shift the state along this continuous variable. These models predict that there should be, in the states of the neural circuit, there should be a topology and a dimension of the states that you measure that matches the represented variable. So if, for example, the head direction circuit is representing head direction, we should expect to see a set of states that correspond in dimension and topology to this variable of head direction. We also require in these models of head direction that there should be an isometry in the representation of head directions. And what isometry means is that equal changes in the direction of the head should map to equal distances traversed in the internal state space. And this is important because if the circuit is an integrator circuit, it means that regardless of the starting position, an increment of velocity should move you by an equal amount starting anywhere on this ring of states. Okay, so this is the thing that I'll call isometry. I just should contrast this with other representations of angular variables. For example, in the visual system, we know that there is orientation tuning to oriented bars in the visual scene. And in that circuit, we know that there is an overrepresentation of cardinal directions, like the vertical orientations and horizontal orientations. So in that case, it need not be the case that just because a brain circuit is representing a circular variable, there should be isometry. Isometry is rather a specific property of an integrator and is required for an integrator circuit. Okay, and the other predictions of the circuit models of the head direction circuit are that all states along this set of angular states should be equivalently stable. So if the system is at a state, it's equally likely to be found on any of the states at any given angle, and there should be no net. If you start the system, initialize the system at some state in the absence of velocity inputs, for example, you should see no net flow or flux along the manifold. Those states should be stable fixed points, and perturbations of the state away from that manifold of states should flow back onto the manifold. So that is the condition of attractive dynamics to a manifold. And finally, you expect that in these models predict that these states should be autonomously stable. So even in the absence of any external cues, these states should be able to be self-sustained. Those states should be generated intrinsically and internally or endogenously within the brain. Okay, so to be able to quantitatively test these predictions in the mouse, we turn to a data set that was collected in the Buzaki lab by Adrian Perash. And this is a data set with tens of simultaneously recorded neurons from the ADN, this readout of the head direction circuit that pipes all head direction information into the rest of cortex and the brain. And the data includes recordings from animals as they're awake and exploring during waking, and also during REM sleep and non-REM sleep. And so we're going to use this data and then try to understand one by one the correspondence between the data and the predictions of these models of the head direction circuit. So the first prediction that we want to test is the dimensionality and topology of the set of representations in the ADN. And so to do this, we developed a method using techniques from the mathematical field of topological data analysis and applied it to neuroscience. And so here the idea is that you take the data as points, like a set of points in the state space of the activity of the whole population of recorded neurons, and look at the structures present in the data at various scales of blur added to the neurons. And we can also separately apply dimensionality estimation techniques that are linear and nonlinear. And so from those techniques, we concluded that in the ADN, the states are consistent with the population states are consistent with the existence of one ring structure, so an object that is fully connected except with the hole in the middle, and just one such object in the whole population dynamics of the system. We also found that the circuit is not does not contain additional dimensions. So what you're seeing over here is, again, the set of states in blue that were recorded across 20-30 minutes of animal exploration in a rich 2D environment. The animal stops, it does lots of things, it moves in 2D, it eats, it grooms itself, it rears up on the walls of an environment. So across all of these rich behaviors, we get these points, these blue points, and then the red points are actually points that are generated by first fitting a one-dimensional curve to the blue points, and then just sampling neural activations as Poisson spikes. And you can see that these synthetic Poisson spikes that are generated are no fluffier, no thicker than the set of recorded data, suggesting that the variance that you see, the scatter in these blue points, is likely explained by simple Poisson fluctuations in firing. In fact, we can look at the cell-cell correlation structure and find that once you condition on this one-dimensional variable along this ring of states, there is no additional structure. So when we look at the correlation structure of just the activations, this rich correlation structure, when we condition out the one-dimensional variable, we see very little residual correlations, suggesting that this circuit is representing this one-dimensional variable and nothing else. So we can also do now an isometry estimation. So that kind of established that the circuit contains a one-dimensional ring. It seems like from all our analyses down to the noise floor in the previous plot, there was no additional structure beyond the one-dimensional ring. So now we can ask about, in detail, what is the nature of angular encoding along this one-dimensional ring. So to test isometry, what we do is we fit the spline to this one-dimensional ring-like manifold that is extracted from the data, and we parameterize along this curve a circular variable in an isometric way, so that equal movements along the manifold correspond to equal changes in angle, and then we extract an unsupervised estimate of the decoded angles represented by the circuit. And what you see here is a coloring of the states by this decoded in an unsupervised way angular variable on the left. And on the right, what you see is actually measured head direction of the animal from LED indicators on the animal's head. And you can see that, and of course, up to an arbitrary rotation and an overall clockwise versus counterclockwise flip. And what you see here is, strikingly, these are, I promise, not just copy-pasted images. These are actually distinct images. I think you'll be able to see there's just one or two spots that actually look different, but these are in fact distinct colorings, and maybe the colorings are a little bit messier on the right with the actual measured head direction. So the point being that isometric decoding is an accurate decoding for head direction. We can quantify this by making the isometric unsupervised decoding estimate here in blue on the top, and in black the curve is the actual measured head direction of the animal, again up to this arbitrary orientation and flip. And you can see here that the unsupervised isometric estimate tracks very closely the actual head direction. So that concludes the sort of argument that it looks like the coding along the states on this ring are isometric in the circuit, which is consistent with the circuit performing integration of velocity inputs. So just to check in over here, we have been able to see that the states are low dimensional, and the states manifolds topology and dimension match that of the encoded head direction variable. We've seen that an isometric mapping between movements along the manifold correspond to changes in head direction. And so now we can move on to test stability of the manifold and autonomous dynamics within the manifold. So the thing that's possible again to do with these states is that the circuit contains its own natural experiments because of the fluctuations in the spiking of the response, spiking response of the neurons. And so we can do things like measure fluxes and flows on the manifold. And what we find is that when there are states that are off manifold, more unidirectionally flow onto the manifold. So these are the large arrows that point towards the manifold from off manifold. And then the net flux at any point on the manifold, across movements for 20 minutes as the animal is running around, there's no net flux in one direction or another at points on the manifold. So this is sort of consistent with the idea that the manifold has equivalently stable fixed points along the whole ring, and states that are off the ring flow onto the ring. So now to the question of autonomously generated dynamics. So here is the set of states that you saw that the animal exhibits during waking exploration in real space. And here is the same animal now recorded during REM sleep. And I think you can see that even though the states might be a little bit more scattered, they still define this kind of convoluted ring in one dimension. And we can superpose these manifolds. So not only is there still a ring structure during REM sleep, but we can, when we superpose those two manifolds, you can see that it's exactly the same manifold during REM sleep as during waking sleep. And this repeats across animals. So in other words, this is a case where the state of the animal is completely different. Its sensory inputs are off because the animal is sleeping, but nevertheless, it generates the same set of states during waking and sleep. Okay, so I think that short of a connectome, but with dynamics, we're actually able to validate a large number of the predictions of these continuous attractor network models of the head direction circuit. And that has been kind of really exciting and compelling that we're able to do this. But we can now start to ask some additional questions. We can start to ask questions about what is the dynamics of states during sleep. So one way to use all these beautiful data from the head direction circuit are, I think, to probe maybe pathological states in circuits, and also different states, behavioral states, and try to understand how dynamics changes across behavioral states, and maybe from normal to pathological conditions, right? And so the head direction circuit becomes sort of the gold standard, the ground truth for how we understand circuits to function in a normal system and in waking. And now we can try to understand maybe sleep through the lens of the head direction circuit, or we can understand things like maybe autism or Alzheimer's or other such pathologies through the lens of the head direction circuit. So here I'm just going to tell you a little bit about the sleep dynamics. And so what we find during REM sleep is that the dynamics, we can look at these dynamical trajectories along the manifold during sleep. And what we find is, first, if we look at the blue curve, the blue curve here is a plot of the change in angle as a function of time, the squared deviation in angle as a function of time. And during waking, because the animal makes coherent movements, it has these trajectories that are consistently moving in a given angular direction. So you get this sort of quadratic increase in the deviation of angle as a function of time. And so, although you can't quite see it, this blue curve is quadratic. I've zoomed in here on this inset over here, and these are coherent motions, so the movement is quadratic. Now, when we look during the REM sleep, this curve, the green curve over here, is actually quite linear, so which is consistent with diffusive dynamics during REM sleep. But one thing that we note is that although during REM sleep the dynamics are diffusive, which are consistent with sort of random walk along the manifold, the slope of this random walk is very large. It's actually moving, it's moving a lot during REM sleep, and so the movements, the amplitude of movements or the amount of movement is not consistent with just stochastic spiking based fluctuations driving random walks on the ring. And so the question that we wanted to understand is what is driving this movement of the state during REM, even though it's consistent with random movements, but it's not individual neuron noise. So what we found through simulating this circuit with the same number of neurons as we understand to be in the real circuit in the brain is that by adding actually random noise, not in the individual neurons, but random noise into the velocity input to the circuit, then we get these sort of large amplitude, but nevertheless random direction movements along the ring. So in other words, random inputs that project into the velocity input to the circuit are what are driving the random walks during REM sleep. And so in other words, what we're seeing is that random noise itself produces very small amounts of, in individual neurons, produces very small amounts of deviation. And so our inference here is that in the circuit, the head direction circuit, individual neural randomness is not enough to destroy memory in the circuit, but randomness in the inputs to the circuit are the dominant source of error in the circuit. So we found this to be very interesting because it's consistent to a bunch of literature in sensory systems. And so this is work from the Steven Lisberger's lab and as well as from Bill Bialik, showing that in sensory systems, the neural circuits are not injecting noise, but rather the precision of representation is limited by the precision at the sensory level. And the same story is true here in the head direction circuit, where precision is limited by velocity noise, but not by individual IID noise in the neurons. And now we can look at dynamics during non-REM sleep, which look quite different from dynamics during REM sleep. So during non-REM sleep, what we find is a second dimension in the response of the neurons. Now they have these pronounced amplitude modulations, which make the circuit dynamics look like, the states look like a cone. So it's the ring and all amplitude modulations of the ring forming this cone-shaped manifold. And what we find is dynamics on the manifold which look more like waking dynamics, which are coherent sweeps along the manifold. So now you see these trajectories that are coherent trajectories. These purple sweeps are kind of sweeping through across a bunch of angles. And then there are these two modes in which either there's sort of local random walking punctuated by large coherent sweeps, and these coherent sweeps correspond to these spindles that are coincident with shortwave ripples in hippocampus. And we know from the larger spatial circuit in mammals, hippocampus and entorhinal cortex, that these sharpwave ripples correspond to replay events in representing spatial position in 2D. So it's intriguing that the head direction circuit has these rapid sweeps during non-REM sleep. All right, I think I'm getting close to the end of time. And so what I want to just say here is that there are two questions that I have that arise from looking at the mammalian circuit now and comparing it with the insect circuit. So one question that Anne has studied beautifully has been, how do you generate in the insect brain high fidelity representations of a continuous variable using very few neurons? And the flip side of that question is, when we look at the tuning curves that in the mammalian head direction circuit, we find that they're very narrow tuning curves. So here's a representative sample of a number of tuning curves extracted from the exact same individual animal whose population dynamics I was showing you in previous slides. And you can see these tuning curves are very narrow, but they're not as narrow as you might expect from theory. So coding theoretic results on representation of one-dimensional variables argue that the optimal tuning curve should be as narrow as 1 over n, where n is the number of neurons in the circuit. In this circuit, if they're of order 1,000 neurons, we're looking at tuning curves that should, according to theory, be 1,000th, of 2 pi. And instead, these tuning curves, though they're narrow, are about 60 degrees in width. And so this is a fundamental mystery of what sets the tuning curve width in the mammals. So one argument has always been, well, neural circuits often multiplex their coding. If a circuit is coding multiple variables, then the optimal tuning curve result changes. So if you're representing a 2D variable or two 1D variables, it's advantageous to have broader tuning curves. But in this circuit, I've shown you that the ADN responses seem to be one-dimensional and coding no other variable. And that really begs the question of, can we get a theoretical understanding of why tuning curves are as narrow as they are? And in the fly, apparently the tuning curves are wider. So I'm very curious to hear what Anne and Barbara might have to say about that. The other question that comes about is, again, the question of topological order. The head direction circuit seemingly lacks topological order, but then when we look at, topographical order, but when we look at the grid cell circuit in mammals, again, this is a top-down view of the placement of grid cells color-coded by their relative firing phases. There seems to be clustering of cells by similar phases. And in fact, not only are cells with similar phases clustered, but they form multiple clusters, and the multiple clusters are themselves organized on a nice triangular lattice in the cortical sheet. So there is topographical order that reflects the tuning curves of the neurons in the entorhinal cortex in grid cells, but seemingly not in some of the head direction circuits, begging the question, I think, of development. So one advantage of having topographical order, I think, is that it's easy to wire up a circuit because nearby neurons only wire to nearby neurons, thereby making the process of specifying the rules for which neurons should wire up together relatively simple. So it eases the burden on the developmental wiring rules. And I think there's a very interesting question of how the mammalian circuit might be wired. And then I'll just conclude by saying that lately we've been working to try to understand these circuits and the origins of these circuits and how these circuits might wire up from relatively little spatial information. And we've been using self-supervised learning rules where there isn't external supervision about where the animal is in space, but the animal simply must use the available neural encoding space to efficiently pack the unique representations about that space in the available neural coding in volume. And so the idea being that nearby positions should be represented by nearby states, faraway positions should be represented by faraway states. And by applying this approach, for example, to the grid cell circuit in 2D integration, we are able to get multiple grid cells, circuits with multiple different spatial periods coexisting within the same circuit. So we get this emergence of multiple modules within the same circuit. I think, Larry, how am I doing on time?
Yeah, you should wind up.
I should wind. Okay, so I was going to talk about this last part, which was the question related to the question I asked Anne about velocity integration and using the same circuits for different types of velocity integration. So the idea is that if you have a a circuit that, here in blue is representing some low dimensional variable, like the head direction circuit over here. The beauty of having a velocity integration mechanism is that if I've got any variable in the external world, this green variable, which is actual head direction, or on the right, a different circular variable, which is some other, variable that changes in a circular way, one could map each of those variables, external variables, to the same internal set of states. And by doing that, one could quite efficiently, with the velocity mechanism, we could just anchor one point in the external world to one point in the internal manifold. And then as long as we have a generalized notion of velocity, we should be able to then use those states in the internal representation for now representing the new variable. And in fact, it seems like grid cells do this, and I'm very curious to discuss in our open forum the extent to which insects do this kind of representation, and how it is that you can map different variables by generating different estimates of velocity in all of these different spaces, and use those generalized velocity signals in a way that allows for efficient reuse of the same internal representations. So I will wrap up over here. I will not talk about how we could do this, but given the time, I'm very happy to discuss some of the things that we can do in the grid cell circuit with these self-supervised approaches and generation of generalized velocity signals. And thanks once again to you for listening, and to people from my group and collaborators who have participated in this research.
Thank you. Maybe you can stop sharing and...
I will stop sharing.
Okay, great. Yeah, I again, maybe we'll start questions. So, I'll just be fanciful, but you might have thought that the mice would dream about turning. And yet your results make it look like that's not happening, right? During REM, I mean.
That's right, Larry. They don't seem to be dreaming about turning in REM, but it looks like the arcs, that we get, the coherent trajectories that we get in slow wave sleep seem to be more consistent with the kind of dreaming of turning. And that's actually when you see the replay events in hippocampus, right? Because that's related to sharp wave ripple activity. So I guess you could think of it that way. I think fundamentally 1D, I'm not sure we can call it replay. I think Michael Hasselmo has made a similar case because, in 2D, there's a very, trajectories are better defined. If you've got place cells, they're sort of unique trajectories that you take in a unique environment. But because every environment maps into this 1D head direction circuit, it's very difficult to imagine that there is a notion of replay in these, in 1D, because you're overloading the 1D circuit with all the environments that you've ever experienced. So it is an interesting question, what's kind of driving these coherent trajectories in slow wave sleep.
Maybe I could ask a follow-up to that. I was curious if there's any relationship to these autonomous dynamics or from the noise to what the animal actually did when it was navigating. So could you look at the structure of the visual inputs or other sensory inputs and make some sense of the structure of that noise? Or is there a relationship there?
Yeah, I think that's a great idea. There's some beautiful work from Mark Brandon's lab just from last year, where he shows that even in the mammalian circuit, and it's actually very analogous to the stuff in the insect circuit, with the ring neurons coming in and biasing, where the bump is forming. So he showed that, when animals are in an environment with a landmark, then in a transient way, the landmark, well, not only does the landmark obviously reset the bump position, but even when you then subsequently remove the landmark, there's a little bit of a trace of that landmark remaining in the dynamics of the circuit. So it kind of retains a brief transient memory of that landmark, and I think the network becomes not completely equivalent fixed points along the whole ring, but it sort of kind of builds a little bit of a dent in the landscape of the ring. Very interesting, right? So I guess in sleep, presumably you want to erase all of that because you have to maintain a continuous attractor with energetically equivalent fixed points. But it would be interesting to see whether early in sleep, there is some, there's some echo of if you've spent a lot of time in one environment with one salient landmark, it would be very interesting to know whether early in sleep that echo is there, right?
I was going to ask, to what extent you think some of the methods you've used, and I was thinking particularly of the osmometry kind of analysis, could be applied to the insect brain. Is it that the data is not in exactly the same form, but I guess there should be equivalents. And how important do you think, how much variance from isometry should be permitted, if you like?
Yeah, no, I mean, I've been really eager to do these analyses in the insect. I think that, we also don't know whether the insect head direction circuit is really encoding only a one-dimensional variable, or whether, I mean, clearly in the fan-shaped body, we know that it's doing, a lot of things from, Larry's beautiful work, your beautiful work, we know that it's, so there's modulation by other variables as well. And it'll be interesting to know to what extent maybe the protocerebral bridge or the ellipsoid body have more of a one-dimensional representation or not. I think these methods can help to get to the bottom of that. I think we're kind of like in the space where we've got, for the insect, we've got the full connectome, and there's so much we can do. We can directly validate mechanism from these models by just looking at the circuit and saying the circuit matches the models, obvious. But then there's also the dynamical level. And I think one of the most interesting things about insects is that, they are, with their small brains, presumably they do a lot of repurposing and switching, neuromodulatory switching based on the same circuit. And I think it would be very cool to understand, that the dynamics could be slightly more rich than just the connection. That's why I'm super interested in this, how much dynamical reconfiguration there is.
Okay, we can move on to the final talk. So our last speaker is Barbara Webb, and her title is Navigation Circuits in the Honeybee Brain.
Okay, hopefully this will work to do the presentation. Is that?
Looks good, yeah.
Good, yeah. Fine, okay, so again, I give my thanks for having the opportunity to speak to you in this very interesting webinar. And I'm going to take a slightly, I think, a nicely contrasting approach in that I'm going to focus on one animal and its behavior as the motivation, rather than the very interesting kind of general approaches, for example, that Anne presented of, what is the principle of navigation here we might be interested in. And the work I'm going to present is I've highlighted 3 of the people who've done the main bits of modeling that I'm going to talk about. There are other people involved as well who will come up as I go along. But what I'm going to talk about is the honeybee brain. And people know that honeybees are kind of famous for being very good navigators. And just to kind of summarize what's amazing about them, a single honeybee has in its head some quite explicit instructions of where food is. Fly 60 degrees to the right of the sun's azimuth for 800 meters, and I'll find the food there. But even more amazing, while it's doing this little dance on the honeycomb in the hive, the bee following this dancing bee gets that information and can now go out and fly at 60 degrees to the right of the sun's azimuth for 800 meters and find the food. So I'm just going to very quickly go through what the key things are that we want to explain in honeybee navigation. So the first one is something that's come up already, this idea of path integration, which honeybees can do over kilometers. So they can fly out from their hive, exploring, looking for food. And when they find food, they can go back home. And just to make sure everyone's kind of on the same page of what we're talking about, we can think of path integration. It's also called dead reckoning when humans do it, that basically you have a set of motion vectors or velocity segments, and you add those vectors all together, and the B line, we call it the B line because bees do it, is the sum of those vectors. And if you increase this resolution, then it becomes a continuous velocity integration. And sometimes people say, well, but this is all very well, but it doesn't work in practice. But it does work in practice for the bee, particularly because they have an external compass signal, which reduces the cumulative error that you would otherwise get. Okay, so what else can they do? So they can memorize, when they found the food, they can memorize that vector from the hive to the food, and later on can go back to the food again with time compensation for the fact that the sun has moved. And they can even remember multiple sources of food and start to develop efficient routes between them. So this is an example actually from bumblebees, but bees have also been shown to do this, that they if there's a few different food sources in the world, they initially will be exploring and discovering them. But as they go round this out again to forage multiple times, they'll end up forming these efficient routes around those. I've put short est was once claimed, but as we know, that's actually quite a difficult computational problem to solve. But there does seem to be at least some indication that they will not they may not necessarily just go to the nearest one. So they will take a route that is shorter than if they just always chose the nearest, for example. What else can they do? They also use visual memory of the landscape to support relocation of home or resources. So this is just one example where bees, for the first time, have gone out on a long flight from their nest. And then you take them and put them somewhere a long way from their nest. And if they've never been there, they're quite lost. But if they've been there once and you've displaced them so they can't use their path integration, they nevertheless can fly pretty much straight back home again. And here's another example. And we have modeled this behavior. I'm not going to talk about this model very much in this talk, but I just wanted to kind of highlight it as we go past. We think that at least one possible explanation of this is that they have view memories that are stored as sparse patterns in this associative area, which is called the mushroom body in the insect brain. And essentially, they have sparse patterns and then a low dimensional readout, which is a familiarity signal, which they can use to recognize when they're pointing in the nest direction and to go in that direction. So I may come back to that at the end, and it may also come up perhaps in the discussion at the end as well. But meanwhile, obviously, the really exciting thing that honeybees do is communicate this vector information to conspecifics. And here's just a little example of a honeybee doing its little dance while the others are watching. And very famously, Carl von Frisch was the first person many people had observed this and even back to Aristotle and believed that it had some kind of communication purpose. But Carl von Frisch was the one who kind of really established a couple of things about this dance. So one is that the angle of the waggle relative to gravity, so this is a horizontal honeycomb, represents the angle of the food source relative to the sun, and the duration of the waggle represents the distance to the food source from the hive. And if a naive bee that's never been to that food source follows the dancers, it's more likely than not when it goes out to arrive at that food source. This was actually controversial for a long time. People said, well, maybe they just know that there's food out there and they're using other odor cues or they're following other bees, etc. But eventually people using radar tracking confirmed that, if you take the food away, don't let the other bees go out. Nevertheless, these recruited bees will search at approximately the right location. So this is an example where the food is here, the hive is here, the food's gone, and the recruit flies out in the right direction and stops and searches at the right distance as well. I say approximately, and this is just one example where this is the nest, this is the food, and these were the first, where the bees started to search. And what you can see is there's a pretty big scatter. Okay, so it's not super accurate, but clearly it's better than knowing nothing about where the food is. Okay, so what happens in the brain? So that's what I really want to talk about today. And it's something that for many, many years, people really had no idea what was happening in the brain. We can now propose quite concrete hypotheses at the identified neuron level for how all these navigation capabilities are computed in the small brain of an insect. And it draws on a lot of neuroanatomy and neurophysiology and including data from Drosophila and other insects, because these circuits seems to be very highly conserved. But nevertheless, all the work that I'm going to present, or most of what I'm going to present, goes rather beyond any direct evidence. So it's not like we have neural recordings from the foraging bee where we see, that these things happen. So we're often hypothesizing. We know what the neurons are in the circuit, but what their function is still a hypothesis that's not completely proven. We benefit from a very, very rich context of ecological accounts, behavioral experiments, and algorithmic explanations of this. But I think what's really important is to say that computational modeling is really crucial to let us connect the neural circuits and the behavior, because we don't have these direct confirmations. Okay, so, and just to kind of summarize or refresh your mind about what Anne said about how the insect brain seems to encode and manipulate vectors. So we can talk about a kind of array of neurons, a topological array of neurons. And if we want to encode a particular vector, we can encode it in the relative, basically as a sine wave of activity. So across this array of neurons, their activity is stronger or weaker, and that represents a sine wave where the phase is the angle and the amplitude is the length of the vector. So I'm just going to, in the rest of the talk, be kind of showing that in this kind of way. And that's nice because then we can do lots of operations on vectors. So if you want to invert the vector, then we just need to have inhibition of the next layer, and we've inverted the vector. If we want to rotate the vector, and Anne already mentioned this, we can do it by connecting with column shifts, for example. So if we want to rotate by 45 degrees in this 8-neuron arrangement, we just do a column shift, and we've rotated the vector. If you want to change, if you want to scale the vector, then we can do that by having some kind of constant modulation across all the columns, for example, to make the input weaker across the columns. And then of course we can do simple addition of vectors by adding together the inputs from 2 columns. So we're going to use all those as we go along. So, okay, so in the bee, in the bee brain, we also have this area called the central complex. And we also have this ellipsoid body, although it's not closed in a circle. And we're pretty certain that it has a compass, and to the most part, celestial cues like sun and polarized skylight are providing the main input. So we have a bump of activity that represents the heading of the bee relative to celestial cues. And so this is an allocentric heading, at least it's with reference to an external reference frame encoded across this array of neurons. But to do path integration, you also need to know your velocity relative to the ground. And a plausible sensory cue for this is optic flow. There's a number of experiments that suggest very strongly that it is the cue that bees are using. And we have input neurons that code for optic flow, in particular, as discovered by my collaborator, Stanley Heinze. So this is just one example, recording from particular neurons in the nodulae of the central complex and giving simulated optic flow, so Star Wars-like patterns going in front of the bee. This particular neuron responds very well to forwards optic flow, not to backwards optic flow, and the response scales with the simulated velocity. So what was very interesting when he examined this in more detail and looked at what's really the preferred for each of these neurons, of which there are 4, what direction of motion do they prefer throughout their visual field? And what you see is that for example, this is a preference for leftward motion and for rightward motion or kind of clockwise or anti-clockwise motion, but the crossover point is not at 0. It's, which you might expect for forward optic flow, it's at 45 degrees or minus 45 degrees. And then it's inverted for these other two. So what that means is that the preferred expansion points are at 45 degrees, plus or minus 45 degrees, and plus or minus 135 degrees. Or we can kind of look at it like this in terms of the preferred response. And what's nice about this is that it actually creates an orthogonal basis that allows the insect to encode holonomic motion with respect to the ground, even if it's not moving in the direction it's facing. So this is the kind of idea. This is the black arrow is the direction that the animal is facing. We have this kind of basis function for these 4 different preferred directions of motion. And if it's actually side slipping slightly, so it's flying slightly to the right of the direction that it's facing, you get stronger input in one of these and less in the other. So you're basically encoding your actual motion. And this was something we proposed based on these results from the sweat bee, but has now been really beautifully played out in recent work in Drosophila, which has included, of course, work that Larry has done as well. And importantly, as we observed, but as they also really nicely demonstrated, this signal, as I've described it so far, is an egocentric signal, but you need to transform this into the allocentric reference frame. And this is what the central complex does for us. So we have our heading signal, we have our translational optic flow signal, which is in this orthogonal basis. So we're getting stronger input. For example, if it's flying this way, we're getting stronger input in these 2 neurons. And what you can do is take your heading bump and make multiple copies of it and modulate the strength corresponding to how strong the motion is in each of those directions. Okay, so the relative optic flow in each cardinal direction modulates the orientation bump, which is this just multiplying by a scalar factor. So we, in our original model, proposed that this formed a kind of distributed representation of the bee's ground velocity. But these later works in the fly have suggested, and I think it's a very viable idea, and it's probably the correct solution, is that in fact, you get these 4 bumps, but then anatomical connectivity does all the offsets that you want, plus or minus 45 degrees and plus or minus 135 degree offsets, so that you sum these all together, and what you get out at the end is the allocentric ground velocity estimate. So here you had the strongest in the direction you were facing, and now the strongest is the direction that you're actually flying. Okay, so you're representing your actual velocity. Both the direction and the strength of the signal gives you your velocity. Okay, so that might have seemed a bit long-winded to get to path integration, but luckily it's now actually completely trivial to do path integration, at least theoretically. All you need to do is integrate your velocity. So you just need some kind of integrative memory. And this was actually proposed as a theoretical model by Wittmann and Schwegler almost 30 years ago now. You basically get, for each segment, you're just adding a different sine wave of activity corresponding to the vector that you're flying. And so the sum that you get at the end is going to be the sum of the vectors as we want. And you can also use this to steer home, because basically this integrated memory is the goal. That's your home direction that you want to go to get back to your goal. And as Anne described, there's a very simple way to do the steering. If you have a goal and you have your current direction, you shift your current direction to the left and to the right and do a comparison to your goal and add up the match, and that will give you steering to get home. And again, this is now being beautifully demonstrated in a number of works on the fruit fly. So I'll just briefly show, this is our kind of, oops, is that going? Yeah, so this is just a simulation of this circuit, which, although I've kind of shown it laying out in this format, it is topologically a ring. in the insect, and we can simulate the bee flying out. This is its heading direction. Here it's accumulating the direction that it's been flying, and then it uses that to control its steering circuit and to steer home. And also this produces a nice little search pattern as well without any additional factor. Okay, so, but let's really push this circuit as far as we can go. So first we want to use it to return to the food as well. Well, there's a fairly simple way to think of how that could happen, is that when you reach the food, you want to store this current integrator state as a vector memory. So you go out, you find some food, and you store a vector memory, and then you go home to the nest. You try and get your path integrator back to 0 using this steering mechanism that I just described. And what do I mean by a vector memory? Well, we hypothesize it's something like a set of synapses corresponding to a neuron that is basically going to interact with the integrator. So you're stamping in the value of your integrator when you're at this point to these set of synapses. Then when you want to go back to the food, you activate this neuron and it inhibits here, so that what you're actually getting as output to your steering is the difference between your current estimate of your location and this memory. Okay, so if you think of it diagrammatically, it's like it's as though you imagine you're here by inhibiting. You then try and get these two things to cancel out, get back home, as it were, to 0. So that means you're going to travel in this direction, but seeing as you're actually starting from home, that means you travel back to the food. And this is a really nice flexible system because, for example, it lets you say you didn't find food here. You don't have to go well, if you stop the inhibition, then your path integration vector will tell you where you are and you can go home again. But you could also use it to do a shortcut. So instead of going home, you could switch to a different previous memory, do the same kind of inhibition trick. Again, you're going to try and fly the vector that cancels out what your current position is and what this inhibition is telling you. And if you fly that route, that would be a shortcut to the food. So we've got shortcuts as well. And we can go take this a little bit further and use it to do the trap lining that I described. So let's imagine that for each new food source you find, you store a new vector memory. So we just have a set of neurons here, each one corresponding to a new food source. And then you want to exploit these. You want to go to somewhere not too far. You want to go around them as pretty much as fast as you can. Well, you can cycle through your vector memories, imagining, this is maybe we're getting to what Ila was talking about, that you're kind of, thinking about what you want to do. You don't actually do any of them. You just go through them all, excluding those you've already visited, and you test out, well, what's the amplitude of this PIVM difference? The one with the smallest amplitude represents the smallest vector, smallest distance, the shortest vector. And so that's the one you should maybe take. Or you could just have a threshold that says, okay, once it's below some amplitude, then that's near enough and I'm going to go to that one. So when we applied this in our simulation, we could get extremely similar behavior to what had been seen in the bees I showed you earlier, that gradually you develop more and more efficient routes as you find the different food sources, and then you try and go to whichever one satisfies this nearest thing. So your speed to get round the foraging route goes down enormously. Okay, so but obviously the key thing, or the exciting thing we want to do is really explain bee dance as well. So how could this vector memory have evolved into bee dance? And this is a kind of hypothetical idea of how it could have evolved based on existing species of bees that have different behaviors that kind of get closer and closer to what we see in the honeybee. So for example, there are stingless bees that literally guide other bees to the food. So this bee will come home and it will fly out and other bees will follow it. Okay, so the lead bee just uses her vector memory and the other bees follow. But then there are other bees where they nest outside, they dance on top of the nest, and their dance is really relative to the sun and they're going in the right direction. So what you could imagine is maybe the lead bee starts her takeoff run, but then she doesn't have to keep going. She's gone in the direction. The other bees head off in that direction. She can come back and tell other bees where to go. You then get these species that actually build nests that are that are vertical. And so the takeoff run in this case has now become something a bit more formalized as a dance, and it operates in this new reference frame, but you can imagine how it might have evolved from the previous one. And then finally, we get the famous honeybees, where the surrounding bees can decode the dance in complete darkness in this new gravity framework. So we think, essentially the dance is a miniaturized, reoriented expression of a memorized PI vector. So that raises the obvious question, well, maybe the follower bee can use their path integration circuit to assimilate the vector. So for example, if they're following the direction that the dancer is going, well, then they could just be integrating while they're doing that following. So that was the kind of hypothesis we started with. But there's a bit of a problem with this, which is that the follower bees don't actually follow. Okay, so if you watch this dancer again, and then look at the followers around the dancer, and hopefully you'll notice that they're all around her. They're going in different directions. Some of them are kind of following the action, but others are just kind of sitting beside. It's extremely variable, flexible, etc. So basically, follower bees do not just follow the dance, but are positioned all around the dancer with their positions constantly changing. And people have shown that they don't need to follow the dance to get this information. So in this work by Anna, we did a lot of high-speed, high-resolution filming of dancing bees and the followers. And what we observed is that their relative angle to the dancer is reflected by their antennal positions. So this is the dancer. These are the several followers around, and you can maybe see already that, for example, this antenna here is bent this way, whereas this one's quite forward, whereas on the other side, it's the other way around. This one's bent and this one's quite forward. And behind, they're roughly at 45 degrees. And this actually turned out to be a very consistent relationship. So this is showing the left antenna, the right antenna, and the center point of the two antennae, and they consistently reveal the angle to the dancer. So what we thought is perhaps the antenna information could be used to calculate the allocentric direction of the dancer in exactly the same way as the bee itself could calculate its allocentric motion, ground velocity, even though it's not heading in that direction. So we're using exactly the same circuit. The bump is now produced by gravity. So the follower bee knows its own direction relative to gravity. And this antennal information is being translated basically into what is the relative motion of the dancer. Okay, so basically we're just taking the angle of the antennae and translating that into activity in 4 cardinal directions that the dancer could be. We modulate the bump in exactly the same way as before. And what we get out at the other end is the direction that the dancer is moving. So, and it doesn't require the follower to stay still. The follower can be changing its own direction to gravity continuously, and changing its angle to the dancer continuously. So its bump is changing, the antennae positions are changing, and the modulation of the bumps are changing. But once you add them all together, what it gets out is this nice steady signal about which direction the dancer is going. And that's what it needs to integrate. So we actually tried this directly. So we took, this is a little video that shows this is the dancer waggling. This is the measured angle of the antennae of this follower. We feed that into this model. So we modulate these 4 copies of the gravity bump, which is the direction that this follower is facing. And we integrate that all the time she's following one of the waggles. So what you're seeing here is the estimate of the direction and then the integration that's happening. And I think this will start again and we'll see a second example. So this is a bee in a different position. Again, the estimate's quite variable, but it's gradually integrating over time. during the time it's experiencing this waggle, and it's getting out an estimate of what the other bee is trying to show. So importantly, the duration of the waggle is getting translated into the length of this vector at the same time. Maybe in the interest of time, I think there's a few more examples, but I'll go on and we can come back and say. But what's lovely about this is basically from the first time since von Frisch discovered what the bee dance meant and could read it out visually himself, we can now decode the foodward vector from the follower's behavior. Okay, so we feed the recorded position and antennae data from real bees into this simulated neural model, and we get out estimate of where the dancer is trying to signal. As I said, we assume the integration occurs while the follower is in contact, which implicitly produces a vector with a length proportional to the waggle duration. And these might look pretty noisy, and they are pretty noisy, and we can discuss that. But bees actually follow repeated dance phases, and people have long suspected that by doing so, they average the information they get to improve accuracy. So when we had, for those bees for which we had data from them following multiple phases, this is one example. This was 3 different phases that it followed. And you can see that the length is quite similar, but the angles are quite spread, but the mean is closer to the correct angle. And if we look at the kind of over all the bees that we could, this is the estimate that we're now getting, the angular error that we're getting for these averaged phases, which I hope you will agree compared to what we saw for the real bees at the beginning, which probably have more than just the angle of the antennae. They're probably experiencing forces. There's maybe other information out there. We're getting quite good estimate of where the food is. Okay, so I have a bonus slide. Do I have time for a bonus slide, Larry? Yeah, okay. So my bonus slide is the time compensation. This is very new work. This is not yet published. It's in under revision. So this is work by my postdoc Evripides. So I've talked so far about how the celestial cues are kind of encoded as this sinusoidal pattern across a neural array, okay, encoding the current heading relative to the sun. And one way you could think about that is you could have a neural array that's basically, it's the cosine of the difference of the heading from the sun direction, or you could have the sine by just rotating 90 degrees, which you can do with the connectivity. So interestingly, if we look at intrinsic clocks, what do we know about intrinsic clocks in brain circuits? Well, they produce sinusoidal oscillations, and you can use these to predict the sun's angle. So this is, for example, mRNA things that are found in neurons in insect brains that vary across the duration of day or the light-dark cycle in a way that looks rather like a sine or a cosine of the time. So this is kind of called the hour angle. And that basically represents the position of the sun as a sine and a cosine. So what Evry proposed was that maybe those two things could be brought together to do another nice bit of trigonometry in this circuit. So we basically modulate the sine and the cosine of the measured sun direction with what we predict the sun direction to be, and we add those together, and what we get is this nice little identity that we're multiplying the predicted direction of the sun and the difference from the measured direction of the sun for the sine and the cosine. You add those together and we get as output what we actually want, which is the difference from north. So this could be a mechanism by which the path integration could actually, the whole central complex could be operating in a geocentric frame of reference, not a sun frame of reference. Okay, so I'll just conclude then. So what I've tried to say is that the structure of the insect central complex suggests a computational mechanism for manipulating vectors in the form of sinusoidal activation across columns. And once we have this insight, then it's actually straightforward to implement an amazing range of things. So we can estimate allocentric ground velocity, we can do path integration, we can do goal-directed steering, we can store vector memories that let us come back to the same place again, or take shortcuts between places, or optimize our route between multiple places. We can use this vector memory as a dance that tells another bee, in this case, how to get to the same place. And we can use the same thing to estimate the motion of the dancer to get a new vector memory. There's many remaining questions, but two that probably are most salient to me are, what is the actual physiological basis of this path integration memory? And the second is, I've sort of said, you stamp in the value of the path integration when you find the food, but what does that mean to stamp it into the synapses? What kind of learning mechanism would make that possible? And then secondly, I hinted earlier at the fact that there's these visual memories. To what extent are these visual memories actually linked to the vector memories? Can experiencing a visual memory revive a vector memory in the brain of the insect? And I would argue that if it can, then we really should call that a map. So I'll finish there and say thank you to the people from my lab who have worked on this, my funders, and of course my colleagues and collaborators. Thank you.
No, thank you. Your talk raises lots of things, but that maybe we'll do in the discussion. But so maybe I'll start with a question for more specific to it. Can you unshare?
Yeah, I'll stop sharing. Yeah.
So in the mind of the dancer bee, there has to also be a kind of a coordinate transformation to relative to gravity.
Yes.
Right. And I mean, gravity is sensed by the antennae, right? So is that, can you relate that to the circuitry you described?
Yes, I mean, we're not certain there's probably multiple places gravity could be sensed. So people also think it might be in the neck of the bee. And we did, for a while, we wondered whether the two things could come together, whether both gravity and the dancer could be influencing the antennal position, but we couldn't find direct evidence for that as far as we could see, but it's still possible. So yes, so it does make you wonder. So I think one key thing here is that one of these input routes is coming through the ring neurons into the ellipsoid body. So it's information about direction is coming in that way, which is setting your kind of compass bump. And then you have information coming in the other way through the nodulae, which is modulating these copies of the compass bump. And I think we already see in the wind system, we see that wind comes in both through the ring neurons and into the fan-shaped body through other directions. So I think there's two different ways that different sensory inputs, or the same sensory input, can enter into the system for different functions here. So my guess would be maybe that's the case for gravity as well.
Barbara, that was like stunning. It was unbelievable how far you've gone. It's really incredible. I just had a detailed question because, like Larry, I have a bunch of questions I would love to discuss as comparative between mammals and insects and how, but I'll come to those when we discuss. But a clarification question is, in communicating with the waggle dance, the distance and the direction, in your circuit model, there's a part where the listener bee or the follower bee has to add up the duration of the waggle to know the distance. But then you said that there's also an averaging across multiple bouts of the dance, right? So it has to switch from adding to averaging. And
yes.
Done.
Yeah. That's a good question, and I don't have a good answer for it.
Okay, so your model has like the adding part, but not the averaging part.
Yeah, we're kind of just doing the averaging offline, as it were. So far, we don't have a good sense of how it's switching. But it's certainly the case. It seems very variable. So the number of times that a follower will follow can be huge, can be between 1 and 30 times they'll watch it. And it clearly they can't be adding up through all of that because they get very different distances. So something has to be happening that lets them change between those two modes. But yes, I don't know how. Yeah.
Barbara, I was wondering, how precise is the dancer bee's dance, and do we know how that is executed? So it's related to Larry's question of sensing gravity, but also in terms of generating the correct duration of that waggle.
Yeah, so I think the short answer, at least from our observations, and I mean, we've gone back to some older data, but also, just analyzed it ourselves to really be sure, is not very accurate. Okay, there's really big, if you look at the angles that they dance, there's really big variation. And in fact, it's actually quite difficult when you say, well, how do you measure what that angle is? And if you go back through, the literature, people have had lots of different ways in which they estimated that angle, and all the early work, it was all done by eye. They'd watch it and put a projector over the, but for example, the dancer doesn't turn and waggle very straight and then turn. It actually starts waggling as it turns. And so where do you start the measurement from and where do you end it? It's quite difficult. But even if you try and use something, quite a consistent way of measuring, you still get very big variation in the directions that they take. Yeah.
Yeah, so let's move into the discussion. I mean, there's a ton of stuff to discuss. That the three of you, thank you very much, by the way, for beautiful talks that raise so many questions. Maybe I'll throw out one, because Barbara, you introduced the MAP word, right? you talked about a map. Of course, in mammals, I mean, Ila's work on grid cells, there's place cells. There's a kind of like a grid type map, or whatever you want to call it. You've talked, Barbara, about a vector map. So how do we think about these two maps? Do insects really do it without the analog of place/grid cells? Is this two different strategies? What's going on here? I'm curious what you all think.
Well, I can start, I guess. So I think the vector system in the insect is maybe not so different from the grid system. So I think it's different in the sense, I think the mechanism is different, but I think what it can represent is essentially the same. I think it's just two different ways to represent similar things, which is essentially how far have you gone in what direction. And I think there have been some analyses that sort of, because it is, people often think, the insect system is polar coordinates, but it's not. It's Cartesian coordinates, just with multiple axes. And one way to think about the grid cells system is that it's lots of little axes, which represent multiple places. So I think there's definitely, in terms of function, I think they're very, potentially very similar in what they can do for the animal, although it maybe has been implemented in a different way. That's how I would think about that. So I think they're both equally map-like in that sense of being able to track in an allocentric way your position in space. And maybe I can go on. So I mean, I brought up this issue of, well, but do you have to associate the vision to it? So I think this is, and that's really coming from a robotics perspective, where, what people mean by simultaneous localization and mapping, for example, is that you can use your visual memory of a location to correct for the path integration drift, and vice versa. You can use your path integration to map, your visual or other sensory experiences into locations, and then you can use those sensory experiences in return to update your estimate of your location. And those two things together give you much, much more precise way to find your way around. So in insects, I think it's been quite controversial, or at least unproven, what extent they can actually do that. So to what extent do they really, from anywhere other than their nest, which is obviously a very salient position, but to what extent, if you put them in a place they've been before, can they recover the path integration coordinates, as opposed to just use the visual view in some much more direct form to tell them, okay, I should go in this direction. It looks familiar there, I'll go that way. That's not using the vector system. So are they two completely independent systems in which neither is, what a robotics person would call a map, or are they linked together, in which case I think they do fit the definition. I don't know if that makes sense, and I'd certainly love to hear other people's comments. Yeah.
I mean, I guess I have a couple of follow-ups to that. I mean, one comment and then maybe a couple of questions. So one comment is that I guess, it seems that the dynamical range could be quite different, right? For this kind of computation, because here with the representing things, with these sine waves whose amplitude and phases are modulated, phase modulation is not a problem, but amplitude modulation presumably has some bounds, right? You can only amplitude modulate over some bound, and so that must put some precision or range limitation. And so I don't know if it's it's just range, because otherwise the mammalian system is hundreds of thousands of neurons, right? Like of order 100,000 neurons for grid cells. And here insects can solve it with so few neurons. And so what gives? Why would the system be so distinct? It's not like they're functionally, as you point out, I think very eloquently, they're doing path integration, both of them, and so they're kind of different solutions to the same problem, but it must be that there is a functional difference given how the neuron difference is so big. And then the other question was, what can one do to get at the question of, is it a map in insects? So if you were to teleport or put in a black box an insect and transport them to a previously visited location, I guess the hypothesis would be that, if they had a map, they would be able to find their way back home. But if they didn't have a map, they wouldn't, right?
The problem there is, as I said, if it's a previous location and they've learned visually which way they should go from that location. So there's a lot of early work that kind of made that assumption, but then people realized, no, maybe they could be just doing it using kind of a view matching strategy. So it becomes a bit more difficult to define it. So for example, what you would want to do, for example, is take them to that place, let them see it, and then close them off again, take them somewhere completely unknown, and then see if they fly the vector of the place they last saw, for example. That might be one way to try and test that hypothesis. But it's actually surprisingly difficult to come up with a really good test. I mean, I think there's a couple of bits of evidence, but not necessarily. But I think I think it's a very good point about the range, or what's the limitations. I don't know if Anne's thought about that to some extent. So
well, I was actually going to ask you a question, Barbara, about this, following up on Ila's question about whether or not there's evidence for patching together multiple local maps. So could you overcome this range limitation by sort of just anchoring to different local maps cued by different visual scenes, and use that to overcome this? And maybe related, do we know about the capacity for visual maps in the mushroom body relative to the capacity for vectors in the fan-shaped body?
Yes, yeah, no, that's a good point. I mean, I think that the capacity for visual memory is much larger potentially than for vector memory, it would seem. Yeah. So that might be, one reason to have both systems, indeed. Yeah. You're muted, Larry.
I got so enthusiastic about what you were saying, I forgot to. Coming back to what Ila said about the dynamic range, you could also imagine that once you get in the mushroom body, amplitude could be used for other things. For example, kind of a voting system. Do I want to follow the wind direction, a memorized direction, whatever? In which case you lose the ability to do lengths. You can keep the angle part. So, you could almost imagine three conceivable systems here. One is the, points on the map kind of grid place cell system. The other is the full vector system, which I think you've argued for, Barbara. And the other is some system where distances and angles are separated. Now, they can't be totally separated because they rely on each other. But, you know, I've wondered whether distance is computed in an independent way somehow, and you use amplitude for voting, sort of. I wonder what you think of that.
Yeah, I mean, I think that's a very plausible, and I think, you know, because an obvious way to do it is to say that you're certain, you know, you represent certainty about different cues, for example, by their amplitude, and you know, so that's a really good way to combine them. So yeah, but then it can't, as you say, then it can't serve both purposes at once. But maybe in some parts of the circuit it's representing length, and in others it's representing, you know, strength of cue or certainty about the cue. Yeah.
Yeah, I think it's clear on the input. It's definitely doing length. It's doing the standard vector thing. It's just once you get into the mush of a fan-shaped body, what's happening, which of course is the big challenge ahead for all of us.
Yeah. So maybe Ila, you can comment on, you know, to what extent have people kind of looked at, I don't know, the capacity in the grid and place cell system?
I think the grid and place cell system, the way I think about it is similar to how, you know, Anne described, you know, a strategy, which is that you build a bunch of local maps and then you patch them together. And so I think that I mean, my perspective on this is that the grid cells locally, when you look at the code, you know, what it's sort of trying to do is separate out, you know, local displacements, you know, how far have you gone, you know, on the scale of 1 meter or 2 meters, because then the map in each module of grid cells, because it's periodic, it repeats. And so it really well discriminates, you know, small movements. But then at the same time, it also contains an absolute code for position. So it's kind of like a displacement code, right? Like the differentials in the code are like displacement codes. But then because there is a high capacity of the grid code, because each module has a different period, and the different periods are not just multiples of one another. And so it means that as one is repeating, maybe at some period, then the next one isn't repeating with exactly the same period, and the next one is repeating with a different period, and so on. And so their relative phases actually encode information about a distance as big as the product of all the periods, in a sense. And so it really contains, with a sophisticated, you know, readout system, it could, in principle, like decode, you know, absolute position as well as displacements, but as a naive decoder could just decode displacements. So, and I think that is, you know, related. And then of course the place cells, I think, are sort of the anchoring, like the intermediaries, of the things that are like, you know, connecting the internal grid states with the sensory cues, external sensory cues, right? And they're just forming like this, you know, this space that allows that connection to be made. That's how I think of place cells. And so, right. And I think that does come back to something that you said, which is that, you know, you should really think about, you know, sort of on the local level where of order 1 meter displacement, maybe we can think about the grid codes as like sort of local vectorial kinds of just, you know, not just displacement vector codes. But on a global scale, they do have this information about, you know, about different, you know, long distance positions or, you know, unique environments and so on. So it's kind of like, as you say, like multiple copies, if you will, of a vectorial code. It's like we take the vectorial code and then we copy it and copy it and copy it and copy it. And then, you know, that's the whole grid cell place cell system.
So Ila, would you say that the increased capacity is primarily to be able to scale the problem up, or is there evidence to do something fundamentally different, and that's why there's such a bigger population of neurons involved in these regions?
Yeah, that is an amazing question. I think it has to do, I think the scale up it's not just quantitative. I think it does allow these qualitatively different things, which is that, you know, just if you just think about, like, if you think about the insect's vectorial representation anchored to home as one map, right? And the surrounding visual cues and stuff is one map, then I really do think of the enhanced capacity of this grid cell place cell system, which in both of your language we can think of as like multiple such patches, right, put together. It's just many maps, right? So, and they need not be contiguous. Like the large capacity doesn't mean that you want to represent a very large contiguous space. It could also just mean that you want to represent a large number of spaces. And then more recently, we've been thinking about, and of course the experimental data has been beautiful in showing that, you can actually make maps of like non-physical space domains. And so, so it's kind of like a substrate for just mapping a bunch of stuff, right? And each one is a, local and separate map, each distinct, but then each map has the same, you kind of know how to move through or navigate through each of those spaces because it's the same type of rules and you're just applying it multiple places.
So how do you feel about the kind of the argument that, maybe the system is, that's what the system is there to do, is, like the successor representation type idea, and that the fact that it's only if you we happen to apply it to space and then it represents space, but it's not inherently spatial, in fact, and maybe even evolved for something else and got hooked and started to be used for space. So it's only when we're using it for space it represents space, but we can use it equally. There's nothing about the structure itself that means it had to be a spatial representation. I don't know if you have any thoughts on that.
I mean, I'm curious to hear what Larry thinks, but I think that seems to be the emerging understanding. I think that is the, that seems to be the case. It does seem to be the case, certainly empirically, that the system is not just representing physical space, it's representing more abstract spaces. But evolutionary origins of which came first are very difficult to answer. I guess on the theory side, we do have, some recent model trying to understand what role grid cells would play in a completely non-spatial context. Like, is there an advantage to having this low two-dimensional representation of other abstract spaces? The experiments have been done in matching 2D abstract spaces, but of course, when we talk about abstract spaces, they need not be two-dimensional. So then the question is, what's the advantage? Is there any advantage of representing, other abstract spaces that need not be two-dimensional using the same system? And, the math suggests that there might be some advantages to just having the structured sort of, as you say, vectorial system where locally you have. So the idea is that, whenever we make a map of any kind of knowledge domain, right? Like say a family tree or, anything, there's nice examples that Tim Behrens had in his model. The idea is just that, you're traversing things. The things that you're traversing, there may be some sort of relations that are meaningful, and you want to import those relations and use them in other patches too, right? Like, if I know son of, it means go down a level in a tree, then that's kind of like a vector, right? And that's useful to translate that vector and use it in a bunch of different maps. So I can do some transfer of relational information by having this sort of locally Euclidean vectorized representations, even if more globally, as Anne's saying, when you patch together multiple maps, you can patch them together in an arbitrary configuration. It may not be a Euclidean configuration, but locally you have Euclidean vector representations, is the idea. But curious to hear what Anne and Larry think about, because they think about these questions as well.
Yeah, I agree with you. I think, you might think it started with space because space is so fundamental and then gets abstracted. We as scientists do all that time. We draw graphs, right? Do that all the time. Unless you want to say something about that, Anne, I wanted to raise a question about numbers of cells, because that's obviously a big issue here. And let me ask you, Barbara, you might know the answer. My impression is, in bees, the mushroom body is incredibly expanded, say, from flies, but that's not true of the central complex, despite the fact that, of course, they're much better navigators. The mushroom body makes sense in the terms of the visual memory, but the fact, if it's true that it's not very expanded, suggests that it's really been built right, by insects and kind of used it that way for many, highly navigating insects and not so highly navigating insects. But is that correct?
Yes, roughly speaking. I mean, Stanley Heinze obviously has been doing really nice comparative connectomics across different species, and he does find some interesting differences. So, for example, he's found a cell that looks very much like the analog of what we propose for the vector memory cell, and which interestingly is not found in flies. It is in bees. I think he still doesn't know whether it's in ants or not. But that's, so there may be, but it seems to me, and as I described it, once you have that basic mechanism, it really takes hardly anything to get extra work out of it. Once you start doing this vector manipulation, you can see all sorts of ways it can be used. And I'm actually quite interested to sort of think, what other nice vector sums could this system be doing, and hooks up. So I think it's just a super efficient solution that's then can be co-opted. But I think you're right, then it is true indeed. The mushroom body is it's something like 100 times the size of Kenyan cells in the honeybee mushroom body, and that's probably not because it's remembering a lot more odors. It's almost certainly because it's visual memory.
Yeah, so, in your talk, I was struck by the fact that, once you have many more cells for head direction, it makes sense that you go in other directions and you get these wiggly attractors and all that. But, you really showed it's only 1D anyway. You really didn't get anything for all that. You have a solution that you can do with many neurons, but that's a little, well, you mentioned that it's a little weird.
A lot of neurons, and I have no idea why, right? And I think we also have no idea what sets the tuning curve within that system. I guess in the in the insect head direction circuit, the tuning curves have to be broad because they actually have to be cosines, right? They can't be any narrower because then the cosine.
So in the compass system, they tend to be a little bit narrower, and then there's actually a cell type that turns them into cosines. But they're not super narrow. They're narrower than cosines, though.
I see. So you have like a pattern formation process that requires narrow tuning, and then there's a downstream cell that makes it wider to be able to do the summation. Fascinating. Yeah.
To do the vectors, and so it really matters.
Yeah.
And I mentioned one thing about the numbers of cells. So way back, this is in the paper that Anne was on, the big connectomics paper, that there were these ideas about coordinate transformations from Richard Anderson and others that, if you have a lot of neurons, you can have any tuning curves you want and you stick them together properly and you get it. But if you want to do it with the absolute minimum, you want to use cosines and sines, essentially, because of the addition formula that you showed, Barbara.
Exactly.
So again, but is that system in the parietal cortex just doing coordinate transformations by an inefficient manner? I don't know.
It's a fundamental mystery. I feel like as theorists, we can't solve why there are those many cells. I mean, it's always been a crutch to say that it's doing other stuff, but if it's not.
So when you solve that, we'll get together again and have another
we'll have another one.
So we could probably wind down. I don't know if anybody has some last words of wisdom.
I wanted to add that I think Ruben Portuguese's work in the larval zebrafish provides a really exciting place to think about the connections across these different species and what things might be conserved and not. And so I think that'll be an exciting piece of work to follow as we think about these questions of why more neurons and what sorts of computational advantages they might bring.
Absolutely. Just for all our listeners, that there's a ring structure has been found in that work. That's seems like the fly. Yeah. All right. So thank you to all of you. It was great. It was a lot of fun and wonderful talks. And we'll call it a day.
Thank you.
Thanks very much, Larry.